Welcome to this explainer. Let's jump right in. Today, we're going to zoom way down into the microscopic building blocks of your cells and then pull all the way back out to see how those tiny components are literally unlocking a revolutionary new era of personalized healthcare. If you've ever wondered how your biology works at the absolute core level, you're in the right place. To understand how that wild future is actually becoming our present reality, we've got to start with a big question. Imagine going to the doctor and getting a therapy that isn't just a best guess, one-size-fits-all pill, but a highly specific treatment engineered precisely for your unique genetic makeup. It sounds like sci-fi, right? Well, it's the absolute central goal of modern genomics. Here's our roadmap for today. 1. The language of life. 2. Decoding the jigsaw puzzle. 3. The Human Genome Project, and 4. The future of medicine. All right, part 1, the language of life and those chemical instructions inside you. Let's start down at the smallest functional level inside your cells, DNA. You already know it's shaped like a twisting double helix, but really think of it as your body's ultimate chemical instruction manual. It holds the exact instructions needed to develop, build, and run virtually every living organism on Earth, including us. And it actually works a lot like an alphabet. Just like letters on a keyboard make up words, DNA has its own four-letter language made of chemical units called nucleotide bases: adenine, thymine, cytosine, and guanine. The rule here is incredibly strict, but super simple. A always pairs with T, and C always pairs with G. The specific order of those pairings, that dictates the entire meaning of your genetic code. Now, when you take all of that DNA together, every single twisting strand and matched letter inside an organism, you get a genome. This is the master blueprint. It has absolutely everything required to build a human body completely from scratch. But the scale of this is just mind-boggling. 3 billion. That is how many DNA base pairs make up the human genome. And here's the crazy part. Virtually every single cell in your body contains a complete identical copy of all 3 billion of those letters, packed neatly into about 20 to 25,000 genes. That's a massive amount of data sitting right there inside you. So how do these instructions actually build you? It happens step by step. Genes don't just sit there, they're active directors. First, an enzyme copies the DNA into a temporary messenger called mRNA. That messenger leaves the cell's nucleus, heads out into the cytoplasm, and meets a tiny molecular machine called a ribosome. The ribosome leads the message and links amino acids together in the perfect order to form a specific protein. Those proteins then build your tissues and control chemical reactions. But if that original DNA mutates, it can churn out abnormal proteins. That disruption is exactly what drives complex diseases like cancer. Which brings us to part 2, decoding the jigsaw puzzle, and how we actually read that genetic alphabet. Now that we know what the genome is, the big challenge is how scientists actually read a microscopic 3 billion letter book. The answer is sequencing. Simply put, sequencing is figuring out the exact, precise order of those A, T, C, and G bases in a DNA strand. By reading it, scientists can hunt down the specific mutations that trigger human disease. The most common way labs do this today is called sequencing by synthesis. It is genius. Scientists use an enzyme called DNA polymerase to generate a brand new strand of DNA. As it builds, it pulls in nucleotides that are tagged with a tiny fluorescent label. A light source flashes, exciting those tags, and they emit a specific color, kind of like scanning a barcode. This tech cranks out short reads of about 125 nucleotides in a row, and it does it billions of times all at once. But here's the catch. Because we can only read 125 letters at a time, researchers are left with billions of tiny fragments. To get the whole sequence, they have to read overlapping segments multiple times to make sure they're accurate, and then stitch them all together. It's exactly like putting together a giant linear jigsaw puzzle. Once that puzzle is solved, researchers can finally pinpoint the exact tiny changes causing a disease. Moving right along to part 3, the Human Genome Project, mapping the blueprint of humanity. This massive puzzle-solving technique was really put to the test in a huge international effort led by the National Institutes of Health, and the Human Genome Project was an absolute triumph. They finished sequencing the genome in April 2003, but you know what's really wild? This huge government-led initiative actually finished under budget, and they beat their original late 2005 deadline by more than 2 years. When does that ever happen? And here's a super interesting fact. The genome they mapped isn't just one person. To guarantee privacy, they took blood from nearly 100 anonymous volunteers. The final sequence is actually a generic composite of several of those people. Even the donors themselves have no idea if their DNA was used. So we ended up with this incredible, representative human genome that's completely free in public databases for anyone in the world to use for biomedical research. All right, on to our final section, Part 4, the future of medicine. Taking this from the laboratory to the clinic. All of this, the double helix, the billions of letters, the global mapping projects, it all boils down to how it impacts your healthcare. In the past, doctors basically only looked at genetics for simple single-gene inheritances like sickle cell anemia. But today, thanks to the Human Genome Project, scientists can actually tackle the really complex diseases that plague us today, like cancer, diabetes, and cardiovascular disease. These involve a tangled web of multiple genes and environmental factors. Now, turning these lab breakthroughs into everyday medical treatments does take time. Getting a brand new genome-based drug through FDA clinical studies usually takes 10 to 15 years. But the future is already here in other ways. Diagnostics and screening tests to spot your specific genetic variations are actively being used right now. And pharmacogenomics, which is the science of tailoring existing drugs to perfectly match your individual genetic makeup, is emerging incredibly fast. It's a total game changer. But even with all this incredible DNA data, we have to stay grounded. Genetics is still just one piece of the puzzle. Diet, lifestyle, and environmental exposures play a massive role in whether or not you'll actually develop most of these common diseases. So, your biology isn't entirely your destiny. Knowing how much your environment and daily habits matter alongside your genome leaves us with this final thought. Your genes definitely write the incredibly detailed first draft of who you are. But do you and your environment hold the pen that writes the ending? Thanks for joining me for this explainer. Keep exploring, and I'll catch you next time.